Today's presentation is HIV Déjà Vu, Optimizing Cocktail Therapies for COVID-19. We would like to thank the American Society for Clinical Pharmacology and Therapeutics, as well as Pharmacometrics Africa, for their generous support of this webinar. Today's speakers are Dr. Michael Dodds, Rajesh Krishna, and Edward Mills. Rajesh is currently a Senior Director in Integrated Drug Development at Certara. He has more than 20 years of drug development experience. His past appointments include leadership roles at Merck, Aventis, and Bristol Myers Squibb. He has extensive experience in development of therapeutics, regulatory interactions, due diligences, and project and program leadership. And he is a fellow of the American Association of Pharmaceutical Scientists and the American College of Clinical Pharmacology. Joining Rajesh will be Dr. Edward Mills. Ed is currently a Vice President of Real World Evidence at Cytel. He's also a professor at Canada's McMaster University Department of Health Research Methods, Impact and Evaluation, and the University of Rwanda School of Public Health. Ed is trained as a clinical epidemiologist and statistician, and he has more than 20 years of experience leading large clinical trials and mega cohort evaluations, particularly on issues relevant to resource-limited settings. Joining Ed is Dr. Mike Dodds, who's currently an executive director at the Integrated Drug Development Group at Certara. Prior to coming to Certara, Mike previously served as a pharmacometrics subject matter expert at Zymogenetics, Amgen, and Quantitative Solutions, which is now part of Certara. Mike has authored more than 20 peer-reviewed articles and holds 2 patents. He earned a bachelor's degree in chemical engineering and biochemistry from the North Carolina State University, and a master's degree in chemical engineering from Montana State, and a doctorate in bioengineering from the University of Washington. Mike, Ed, and Rajesh, welcome to today's event. I'll now turn it over to Rajesh to begin the presentation.
Thank you, Suzanne. What I'm going to talk about today in the component that we have is to lay some of the background foundational similarities between SARS-CoV-2 and HIV, and specifically what lessons can be learned from this experience. Trying to advance to the next slide. Yes, there it is. Before we proceed in this discussion, I wanted to highlight a few key resources that would be helpful for the audience. One is the COVID pharmacology website, which is very easy to remember, covidpharmacology.com. And within that same resource, we have the viral kinetic model, which is in part of the in silico workbench. And finally, the crux of today's presentation is being published by British Journal of Clinical Pharmacology. The DOI of the paper, a preprint, is shown.
Yep, there you go.
Great. So we talked about the resources, and now we can advance to the next slide. Great, thank you. One of the things that we want to point out is early in this pandemic, we have seen an increase in the clinical trials looking at potential treatment of COVID-19. And what we see is that early trials have focused on monotherapy, whether it is hydroxychloroquine or remdesivir, repurposed molecules looking at monotherapy and the effect of monotherapy on COVID-19. Some of these trials have been randomized controlled trials. The others have been non-randomized clinical trials. And what we see is that as the trials have progressed with time, there has been a shift in from monotherapy to combination trials. And that's what we see here. These are published reports. The most prominent of the combination is a triple combination study of interferon, lopinavir, ritonavir, and ribavirin in patients admitted with COVID-19. So there's a trend, at least, that early in the pandemic we have seen monotherapy, and more recently we have been seeing combination trials. Next slide, please.
There you go.
Thank you. So I just want to point out a few things. One of the endpoints we look at in virology is viral load reductions. And what we see on the left-hand side is a publication in Lancet looking at remdesivir as a function of monotherapy in comparison with placebo control. And what you see here is whether there's the upper panel or the lower panel, there's really not much effect on viral load, which is similar in the early days of HIV, where the reduction in viral suppression was pretty modest. On the right-hand side is an unrelated work by Hung et al. In Lancet 2020, looking at triple combination. And in this case, the test treatment is the triple combination of interferon, lopinavir, ritonavir, and ribavirin versus the control arm, which is lopinavir, ritonavir. And what you see here, at least marginally, there seems to be somewhat higher effect on viral load. Again, this is not a direct comparison, but at least there's a trend that if you move from monotherapy to more combination therapeutics, you probably would be able to tweak the system to see a more meaningful viral load reduction. Next slide, please. So what do we know about COVID-19 so far? In terms of natural history, we do see some commonalities with other viral cell cycles, in particular HIV. And why is it similar? The process of entry into the cell, replication, and forming an infected viron are similar, regardless of the specificity around the virus. What is very interesting so far is that monotherapy appears to be either not effective or somewhat less effective, modest at best. And again, some of the challenges in the field right now is these monotherapy treatments have been repurposed molecules and given without regard to severity of disease. So where in the process of infection are these compounds administered is also a critical input. Preliminary data appears to point towards incremental efficacy with combinations. And what we are proposing in this presentation is this is something that Mike Dodds later in the presentation is going to talk about, is understanding the viral cell cycle opportunity points. And what do I mean by that? What I mean by that is in the viral cell cycle, there are very specific transition points between one phase to the other. And this is considered an opportunity point. It is also important to understand where in the infection cycle, whether early, mid, or late, can these designer therapeutics be applied. So what we'll do in the next couple of slides is just go back in time to look at the evolution of HIV drugs and the treatment strategy that was pursued at that time. So if you recall, and this obviously is a simplistic depiction of somebody, let's say if you were in virology and HIV in particular, you would recognize some of the challenges that we are facing now as it relates to SARS-CoV-2. Since the discovery of HIV as a cause of AIDS in 1983, there has been significant efforts expended to treat HIV. And that started with the approval of zidovudine AZT in 1987. And what you see on the left-hand side are the various mechanism of action of these compounds. And on the right-hand side of the table, you see the evolution of the treatment landscape. And what is very interesting here is, like before and like now, the early treatments focused on monotherapy until about the mid-90s, where there were approval of the protease inhibitor and the non-nucleoside reverse transcriptor inhibitor. And that afforded more variance in combinations. So as we go down, we see more diverse mechanism of action in play. So what started off as monotherapy continued as within-class combinations that produce modest additive effects, but then progressed to cross-MOA treatments, and finally leading up to PK enhancement. So the field is still evolving, but the standard of care today for HIV is combination therapeutics. If you go to the next slide, this continues in the same vein of the HIV landscape, beginning with the approval of zidovudine in 1987. Couple of challenges relating to that were dose-limiting toxicities and incomplete virological suppression. This is consistent with what we see with remdesivir today. But unlike SARS-CoV-2, where we don't have data yet, HIV saw the emergence of resistance mutations due to the incomplete virological suppression. What is more important in HIV, which could have relevance today for SARS-CoV-2, is within MOA class combinations. How, when you put two compounds together, what are the effects? Is it additive? Is it antagonistic? Or is it synergistic? My colleague Ed Mills will be talking about it in more expanded way in the next segment of this presentation. But in early days of HIV, when you give D40 and AZT, the combination was antagonistic, whereas AZT and 3TC were additive. A couple of days ago, we got the FDA press release showing that hydroxychloroquine or chloroquine, when combined with remdesivir, reduces the antiviral effectiveness of remdesivir. That's an antagonistic effect. So those experiments are obviously relevant and important, and as lessons learned from the HIV therapeutics. Mid-90s saw the approval of PIs and NRTIs, and that allowed cross-MOA class combinations. Heart regimens give you either a combination of two NRTIs plus a PI or an NRTI, and the standard of care is two NRTIs plus a potent third agent. Nevertheless, the focus of the state of the art within the HIV landscape is combination therapeutics. Next slide, please. So in this slide, on the right-hand side is a very nice picture from a publication by Aaron in New England Journal of Medicine back in the mid-90s. What shows here is the viral load reduction on the log scale on the left-hand side, on the y-axis, and time in weeks on the right, on the x-axis. This is very relevant for combination therapeutics. Viral replication is a multi-phase process. The goal is viral suppression. As I mentioned before, there are transition points within the cell cycle, and this is something we'll cover later in the presentation. And what we see here is in the graph on the right-hand side, when you give AZT and monotherapy, you see some modest reduction in viral load. And similarly with 3TC, you see a little better than AZT. But when you combine the two, either in a low-dose 3TC format or a high-dose 3TC format, you see a pretty meaningful reduction in viral load. So I think the opportunity for eliciting a synergistic effect with combination across MOA is a critical consideration, something we should apply within SARS-CoV-2 as well. Now, given the maturity of the landscape, actually, if you go to the previous slide, Suzanne, given the maturity of the HIV landscape, combinations allow you to individualize treatment, whether based on efficacy, adverse effects, drug interactions, or resistance. And those are some of the considerations at play today with SARS-CoV-2. So if you go to the next slide. A note on regulatory guidances. There is obviously an FDA combination rule where each component of the combination should have its own efficacy. In addition to that, there are two other relevant guidances, one on HIV drug development, which was put forth in 2015, and where some of the questions around antagonism, additivity, and synergy was spelled out very well. And this is something that is relevant in the context of the FDA press release on remdesivir. On the right-hand side, there's also a co-development of two or more investigational drugs and combination guideline that came in 2013. And that provides for more specificity around combinations intended to treat a serious disease. Where the second bullet is very telling of the state of play in SARS-CoV-2. You need a strong biological rationale for the combination. But more importantly, within the parenthetic statement, the agents inhibiting distinct targets in the same molecular pathway or steps in disease pathogenesis. That's the critical play. That's the cell cycle feature, which we'll talk about as we go down the presentation. Next slide, please. So, and while I talked about the FDA guidances, there are similar precedences set by other health authorities in various regions of the world, which for time reasons, I would not be covering here, but we will be happy to entertain questions at the end if needed. So what do we see similarity-wise, COVID-19 today and HIV from the mid 80s to late 2000s? The evolution appears to be the same, more or less. We also know that monotherapy appears less effective, and that's what we see today, as we saw before. Viral cell cycle considerations are critical, and that's going to be the subject and theme from today, and we'll continue to the next few slides with other presenters focusing on those aspects. The lessons learned from HIV therapeutics have a direct and proximal impact to the current pandemic. So I think, how do we treat the various transition points, but also how do we stage the severity of disease are the two things that we should think about when designing these therapeutics. Furthermore, the drug approval precedent landscape for HIV therapeutics does play very well into COVID-19 landscape as well. At this point, I will transition to my colleague, Ed, who will take you through the combination treatments and clinical trials.
Great, thank you very much. And good morning, good afternoon to everybody. I'm going to talk about clinical trials in the context of evaluating their therapeutic efficacy, and then it'll give you an update about what's happening in the field, some of the concerns that I have and that others share with me, and how we can build upon the infrastructure that is quickly being developed in clinical trials to get to a better understanding of how we're going to evaluate combination treatments. I have an executive summary there. We can get it. I don't need to go over it. Let's get straight into the slides. So the information that I'm going to be talking to you about comes from an online clinical trial tracker, which is available to you at the web URL at the top there, covid-trials.org, and several combinations of words like that will get you there as well. That is hosted by both Cytel and Certara, and that's an up to the day accumulation of registered trials, clinical trials, the majority of which are randomized trials. But we sift through all of the major clinical trials databases to find out what's registered, what's currently recruiting, what has shut down, and you'll see in the next few minutes about why it's important to be up to date on what's happening with the trial. So as of yesterday, there were 1,476 clinical trials currently registered, but in reality, I think we're going to find it's much, much smaller in terms of trials that are going to provide information to us that is useful. Next slide, please. When we're talking about COVID and clinical trials, I think it's important that we understand the disease stages of COVID. Many of you will have been familiar that 2 days ago, there was results out about a corticosteroid for COVID, and it got a lot of attention, and people were sharing this on social media that there's a treatment that works. It came from a trial called the Recovery trial. When you start looking at that data, and of course it's not published, but when you start looking at it, you find out that's in the late stage and palliative care population, and therefore the findings of that steroid don't make any difference for the other populations, pre-exposure populations, post-exposure, early treatment, and early hospitalization. Similarly, yesterday there was an announcement that the Solidarity trial was dropping the hydroxychloroquine arm and that it was ineffective within a large multinational trial called the Solidarity trial. That's also in the later stage, hospitalized and later stage populations. So we shouldn't infer from that the findings of that drug lack of effect have any role to play in pre-exposure, post-exposure, and early treatment. We still need to evaluate them. But what you're going to see here is that of the large number of trials that were registered, we have about 6% are planned or recruiting for pre-exposure prophylaxis, 4% among post-exposure prophylaxis, which means you've likely been exposed and you're trying to prevent it, the exposure from turning into an active infection. About 10% of all clinical trials look at early treatment. That's where I think we should be really focused on, because early treatment hopefully will prevent people from progressing and needing hospitalization and therefore preventing death. We've only got 10% of trials looking at that. We have about 58% of trials are in the hospitalized population and 14% of trials in the very late stage in palliative care. So as we're talking about trials, or even as you're thinking about the COVID populations that you're interested in, perhaps it'd be useful to divide it into those different populations, because what happens in one population is not necessarily transferable to others. When we look at the 1,476 trials that are registered to date, 87% of them are monotherapies. This is really quite an astounding finding. So they're usually two-arm trials. They're usually quite small trials, less than 100 people, and they're usually only comparing a single agent versus a control. Why this is particularly concerning is that there are certain drugs that are overrepresented. In particular, chloroquine and hydroxychloroquine are very overrepresented, particularly in the monotherapy trials. More than 250 trials incorporate one or both of those therapies, and 112 of those trials are looking at these therapies either alone or versus placebo or a standard of care. Next slide, please. When we think about combination treatments, we find out that only about 13% of all trials, 190 of the trials, are looking at any form of combination treatments. And then there's that hangover effect that I was mentioning in terms of hydroxychloroquine is entirely overrepresented. So 60% of all combination trials involve some form of chloroquine-based therapy. And only less than 2% of trials have been designed in a way, I'll refer to it as factorial, like the figure that I present there, where you could evaluate what are the effects of the individual components of the trial and look at whether or not combining the treatments offers these favorable effects above the individual components. Next slide, please. So when I'm talking about providing combinations and evaluating whether or not these combinations offer anything in addition to monotherapies, we often will use terms additive, antagonistic, or synergistic. And simply what I mean by this, if I talk about additive effects in its most simplistic way, would be that we want to know the effect, let's say the relative risk reduction of drug A, and we know the effect of relative risk reduction of drug B. Therefore, we could make an assumption if you put drug A and drug B together in the same patient, that the effect of it would be equal to the effect size of each individual drug. That's what we refer to as additive. If we were talking about the word antagonistic, then we know the effect of drug A, we know the effect of drug B, we would hope them to be additive, but you put them together, you evaluate it in trial, and in fact, it negates the treatment effect of each drug, and perhaps that's caused by something like a drug interaction. Finally, and the one that I think we're most excited about, or most enthusiastic about, let's say, is synergism. The idea that if you give drug A, which you know the effect of, and you give drug B, which you know the effect of, combined, they offer you better than an additive effect. And this occasionally does happen. Next slide, please. Now, where this becomes challenging is if we start looking at multi-drug components. So for example, 3 drugs, which is what we use usually in HIV treatment, then that doesn't mean that the 3 drugs are all going to be equally additive or equally antagonistic or equally synergistic. You may have to be very careful about choosing your drugs, as you see in the figure to the right, because 1 or 2 of the drugs may be additive, and you add a third and all of a sudden it's antagonistic and you've reduced the additivity effect. Or you can imagine 1 drug is, 2 drugs are synergistic, but you add another drug and it's antagonistic. So choosing and evaluating whether or not an additional drug is exerting a therapeutic effect, whether that's beneficial or negative, is something that we need to determine from clinical trials. It's only once we get into trials we're going to be able to figure that out. In HIV, the next slide illustrates this. There is a synergistic effect. And of course, those of you who have lived through the HIV epidemic will remember AZT monotherapies, the days of early 90s, where it was monotherapies. They tried out paired therapies. And what you'll see from this AIDS clinical trials group study, the 343 trial, which was a very important trial, they showed that if you took the individual drug and compared it to the paired drug, so indinavir versus zidovudine and lamivudine, which was being tried at the time, indinavir alone was slightly better, but probably not importantly better. When you combined all three, it was better than either the monotherapies or the combined therapies. And so it offered some form of synergism. And that's how we practice HIV medicine these days. And we go from having very low rates of viral suppression. You see there individually, they offered 55 or 60% viral suppression. You combine them and you have almost 100% viral suppression. And that's, thankfully, HIV has become a much more manageable disease than it used to be. And we see the same thing with tuberculosis, and we see the same thing with hepatitis C. Combined drugs appear to be the way to go forward. Next slide, please. In terms of COVID, combination therapies, as they currently stand, can be quite difficult to interpret. So there was this paper by Hung in the Lancet several weeks ago, about 5 weeks ago now. I thought it was a very important study when it came out because it showed the combination treatment of lopinavir, ribavir, and interferon was better than lopinavir alone. It was a very important study because it showed a treatment effect in hospitalized patients that was significant, meaning some drug is going to work here. The problem is we can't tell which drug is working because they compared lopinavir alone, which we don't, as their control group, which we don't know the treatment effect of compared to nothing. They compared lopinavir alone to lopinavir, ribavir, and interferon, so a triple combination. And so you've got a combination treatment compared to a monotherapy that you don't know the individual effect of. And therefore, although we can interpret there is a difference between the groups, we don't know what is driving that. Is it lopinavir? Is it ribavirin? Is it the addition of interferon? We simply have no way of interpreting it from this study. If we look at the figure on that slide, the figure on the right-hand side at the top, that's probably the most important component of this trial. That was the nasopharyngeal viral load swabs. And while there's definitely a difference, they had 7 days of treatment. If you look at this, you have the blue is the control group. I think, yeah, blue is the control group, and you have red as the combination treatment. But what's interesting to notice here is that the difference appears to occur between day 0 and day 1. And after that, the slope of the treatments are remarkably similar. So although we have this interesting paper, looks at the topic we're interested in, combination treatments, and comes out in the Lancet, at the end, it's clinically not very useful. We don't know what's going on here to benefit the patients. Next slide, please. I mentioned to you antagonism. So a couple of days ago, the FDA put out a warning that hydroxychloroquine, which is becoming, may or may not have a role to play within the different populations we looked at, may interact based on in vitro data with remdesivir, which is now one of the recommended treatments for hospitalized patients. And so although this comes from in vitro studies, they put out a recommendation that perhaps these drugs shouldn't be used together, but it doesn't come from randomized trials. It doesn't come from trials where we're able to go and determine the reality of this. So it's an example of the expected antagonism that might occur, but of course, this doesn't come from a high quality source of evidence. Next slide, please. I think one of the solutions to this is the new approach to doing clinical trials, what I refer to as master protocols. You'll see this written about. There's various types of master protocols, but the type that's displayed in the figure to the right tends to be the one we're seeing in the COVID epidemic. What's interesting here is that only a few months ago, platform trials were a real rarity. Master protocols were a real rarity that only very sophisticated groups that were very well funded were doing. And only a few months later, they have become the new highest quality, probably, trials that we're seeing in COVID. And any of the findings that that are actually changing clinical practice tend to come from these platform trials. At the moment, the important platform trials that I tend to be following are named the Discovery, the Recovery. Both of those are from Oxford in the UK. The Solidarity trial, which is the WHO trial that you may be reading about, and another one, very important trial called the REMAP-CAP. And why I think the REMAP-CAP trial is so important is because it was already going on. It was a trial, a multi-arm trial evaluating interventions for community-acquired pneumonia. And they pre-planned to have a viral pandemic arm that they could activate in the context of a pandemic, and they did have to do it. So they were remarkably, they had remarkable foresight and clever design, and I think REMAP-CAP will go down in history as being one of the most important trials, particularly from COVID, but maybe in the history of clinical trials. Essentially, what a platform trial does is it allows you to start with multiple arms. Then you look at them using pre-planned interim analysis, and should you come to the conclusion that you can make a decision to do something important, you could drop an arm, for example, or you could add an arm. What you can also do in the context of combination interventions is you could do monotherapy arms and then stepwise combination arms and evaluate whether or not a drug is exerting an additive effect or a synergistic effect or an antagonistic effect. Next slide, please. So to conclude, I'm going to leave you with a slightly different concept, and it's one that's grounded in the reality of how I see clinical trials today, June 18th. I don't think that most of the trials that are registered will ever recruit patients, or they will recruit a few, or perhaps about 10% of their planned populations. The vast majority of studies are not well funded, and they take an awful long time to set up, get the infrastructure, get the training going, get the swabs so that you can take care of patients, or enroll those patients. It's very difficult, and by the time that the approvals have all come through, we're finding there are actually very few patients around that are either willing to be recruited into a trial, or they're just not in the community any longer because the epidemic has moved on somewhat to another location. As a result of this, many trials won't have been able to hit their planned recruitment rates. And I think many investigators will never publish their data because they think, well, who's going to be interested in a trial of 10% of the planned data? I think as a result, many funders, investigators, and patients' time would have been wasted. But I think there's a solution to this, and that is that we need to create some form of sharing the data and creating a data hub where investigators can still share that small amount of data or raw data that they have collected and contribute to building complete trials. If we have 10 trials that only recruited 10% of their populations, if we could get that data, all of a sudden you do have a completed full 100% trial. And conceptually, this would not be very difficult for analysis, but this would, to my knowledge, be the first time investigators actively sharing complete data sets. And finally, if this can be done for COVID-19, it could conceptually be done for all other diseases that have ever been evaluated in trials. So thank you. That's the end of my presentation. I'd like to hand it over now to Mike Dodds.
Thanks, Ed. For the remainder of the hour, I'll walk you through some of the modeling and simulation activities we've been performing to rationally design drug combinations for COVID-19. At this moment in time, no single agent has presented itself as capable of arresting SARS-CoV-2 life cycle. And maybe that shouldn't surprise us. These are, after all, repurposed drugs. They were never intended to treat coronavirus of any kind. And perhaps we shouldn't find that terribly surprising. So our hypothesis here is we should learn from our experience with HIV and, as Ed said, TB, to look at combinations that target different segments of the viral life cycle. And if we do that, we might be able to generate sufficient additive cooperativity so that these drugs that have otherwise lackluster monotherapy performance together might get us over the hump. How would we do that? Well, we've got a viral life cycle model that provides a pharmacologic framework to guide those combination selections. And it also innately handles the issue of time. As Ed commented, the stage of the disease can be quite important. And our hope here is this framework allows us to rationalize combination selection. The combinatorics of the number of combinations that we can attempt is just huge. And so we hope that this modeling work would provide a way to narrow the field down to a handful of combinations that we think most likely. Next slide, please. The model that we're using comes from Antonio Goncalves and Jeremy Gage over the INSERM. It's been published in 2020. It derives from the Bachem 2006 model, which differentiates itself from other infectious agent models in that it assumes a rapid pathogen timescale, and it doesn't then deal with the relatively slower timescale of the infected cell population. It's informed by a relatively low number of subjects. So as we build more information, we'd like to refine this model, obviously, and redo these simulations. While this is sort of the formal way to look at the model, I don't think it's particularly helpful for non-specialists. So let's look at this in a bit more pictorial sense. Next slide, please. There we go, great. So starting out in the middle of the diagram, we have V, or the viral load. So these are viruses that are in the environment, in the lung, the upper respiratory tract. And those virions, V, can go on to do one or two things, right? The thing that we hope for is to be killed, right? And the rate constant at which they're killed is defined as C. The way we might accelerate that process would be to bring in, say, antibodies, or perhaps convalescent plasma, right? Or perhaps a manufactured antibody that can bind up those virions and actually remove them from circulation and kill them, right? So that's the thing that we hope to have happen. The other thing that the virions can do, and sort of proceeding left and down on this diagram, is to find an uninfected host cell, in this case, the epithelial cells in the lung and airway lining, and then infect those cells. And that happens at a rate defined by the term beta in the model. And there again, there are drugs that we think could inhibit that infection event. Once the cells are infected, they become this latently infected population, I1. Those cells are infected, but they're not yet producing new virions, right? Because it takes some time, for example, for viral uncoating to happen and to spin up and usurp the machinery of the host cells to start forming more copies of itself. The transition time that it takes to go from latently infected to productively infected, I2, is governed by the rate constant kappa. Once we get to infectively productive cells in the lower right corner of the diagram, two things can happen to those cell populations. The thing that we hope for is for those cell populations to die, right? To program cell death, apoptosis, for those cells to stop being little viral productive factories. Drugs that might do that, we might think of azithromycin or interferon effects where you actually increase programmed cell death behavior. The other thing that can happen from these infectively or productively infected cells is the thing we don't want to have happen, is to release new virion into the wild, and that happens at a rate governed by the term rho. And things that we might do to drug that particular behavior, that would be your classic antivirals, so protease inhibitors, things that make the cells less effective once they're infected, less effective in producing more and more virions. And so then virions go out into circulation again, and we can repeat this process. The cycle can continue counterclockwise until we either run out of virion, V, or we run out of host cells to infect, T, right? And at that point, the infection begins to collapse under its own accord. Next slide, please. So that diagram is nice. Let me walk you through what that model, once implemented in practice, let me walk you through the model around what we think a natural infection history looks like according to this model. So starting in the upper left panel, we're looking at a time zero. We have a large pool of uninfected epithelial cells, T. And the infection begins at time zero. And for the first 6 days of infection, that's on the x-axis, not much happens to that cell pool. There are cells that go on to become infected, and that's in the upper right panel. These are the so-called eclipse phase cells. In the lower left panel, those eclipse phase cells then go on to become productively infected. And that productively infected period peaks at around day 9. Not surprisingly, then in the lower right part of the panel, you can look at varying copies per mil, and that also peaks, we think, at around day 9 after infection. Then we sort of can't really detect virus. Usually the lower limit of quantitation is around 100 copies per mil. So we really can't even detect virus in these samples until maybe day 5 of the infection. And then we're most often able to detect virus in these subjects out to maybe day 21. So that's the natural history. And as we walk through the simulations, I'll keep presenting the natural history as black lines. So that's a lot of information on those slides, and we start thinking about intervening in this viral life cycle. We need a way to take that information I just showed you and summarize it into some kind of metric that then we can then use to do some apples to apples comparisons. What if I do this versus what if I do that? How does that impact the outputs that we just saw? So we need to collapse that time and concentration information down to some usable metrics. So the first easy one to think about is the viral load area under the curve. You would simply take that viral profile that I showed you coming up, peaking at day 9 and falling off and disappearing by about day 21, and we'd simply integrate that with respect to time. And that reflects then the total exposure, if you will, of this particular patient to the viral load. The next way to summarize these interventions would be around the duration of viral shedding, and it's highly related to the previous metric. But that duration is essentially the duration of time, the number of days for which we can detect virus, and usually that's around 100 copies per mil, because that's the lower limit of quantitation for these quantitative PCR assays. So we think that's an important proxy for things like length of stay, or the amount of time you need to isolate these subjects, because presumably they're shedding virus that's detectable, they're infectious during that period of time. And so if we can shorten that window of time, this could have an important impact on the say, hospitalization duration. And then importantly, from a population health perspective, it should affect transmission dynamics, right? There's just fewer days at which the subject then can pass this infection on to another individual. And so the disease progresses. So that could be a very important metric for population health. Finally, the third metric then is looking at epithelial cells that are infected. And there we just literally look at those epithelial target cells that are infected by the virus and take a look to see what an intervention might do in terms of reducing that number of infected epithelial cells. And that we think is an important metric because that could be a proxy for pulmonary signs and symptoms, right? That as that cell pool becomes infected and is killed off, this is going to recruit host cells and recruit host inflammatory responses. And those inflammatory responses seem to be what responsible for ARDS and later cytokine release syndrome. So walking through the primary findings of the simulation results, each of those endpoints I just talked you through are given here in rows on these plots. Viral load at the top, epithelial cells infected in the middle row, and then final duration of viral shedding on the bottom row. And what I'm showing you with these vertical columns in the panel is time of treatment initiation. So on the far left column, that's initiating treatment 6 days before peak, right? So these patients are asymptomatic. This is something like a pre-exposure or post-exposure prophylaxis intervention, right? That's very, very early intervention. As we move to the right, the middle column, that's intervening right at the time of the peak viral load. And on the far right column, that's intervening 6 days after the peak viral load. So if you just step back from this diagram for a minute and look at the big picture, what you can see is that the curves and their deviation from zero, and the zero line at the top being here the null effect, you can see that these curves begin to die out as we move forward in time. So no matter what endpoint we look at here, earlier treatment is better, right? That what we find is for every single one of these metrics, earlier intervention seems to be better for any of these metrics that we consider. So that's the first primary finding, and I think other researchers have reported on that too, so that's not particularly unexpected. One, I guess, somewhat unexpected finding that we got from this is that some of these metric improvements are time-bound, right? That there are periods of time after which you really are not able to do much for certain portions of the model. For example, starting on the top row where we look at viral load area under the curve, once we get to peak viral load, most of that area under the curve has occurred. So even if we intervene, we don't meaningfully move that metric very much. I think probably more importantly, on the middle row, when we look at epithelial cells infected, we can clearly see that if we intervene at peak, say peak plus 3 days or later, we can't spare that epithelial cell population, right? All the damage has been done and those cells have been largely infected. So there's not much we can do for that individual patient's epithelial cell infection. In contrast, durational viral shedding, the final row on this table, we do seem to be able to do something. Of course, earlier is better, but we can always shave off a little bit of time from the duration of viral shedding. And again, that may be important from a population health perspective. Next slide, please. Probably the finding that we're most excited about, and we're spending some more time investigating that, is we can find evidence that if you take a specific amount of drug intervention and divide that up instead into multiple portions and attack multiple portions of the viral life cycle, you can get a better response than the individual monotherapy. So going back to Ed's comments on synergy energy, we do detect in the model that there's potential for synergy by simply divvying up the drug effect to different portions of the viral life cycle. And this is important because it's unlikely we're going to get any particular therapy that has a particularly fantastic response. So perhaps putting together a handful of these drugs in combination would be worthwhile. Next slide, please. So when we arrange the findings of the simulations by in 5 different ways that we could target the viral life cycle, and that's the columns of this table, and then we score those interventions by the different endpoint metrics that I walked you through earlier. Those are the rows on the table. We can find that the different interventions are preferential. Here, negative means we really didn't see much of an effect relative to the other choices for intervening at that particular point in the model. Plus is best in class. That was those simulation results seem to be they give you the best possible response. And then plus is yes, we got some positive benefit there. So I'll direct your attention to the far 2 right columns of the table. What you can see there is sort of pluses all the way down the board there. So what we suspect from the simulations is that if we target the increasing turnover of the productively infected cells, that's delta, or we target decreasing productivity of those cells, that's rho, of these infected cells, this is predicted to have positive benefit for all metrics. So that would suggest to us that intervention combinations should have components in them that target delta and rho, and those should be the backbone of any proposed combination. Again, we don't know which of these endpoints, which of these rows are the best thing to do for either a population or an individual level, but generally speaking, interventions targeting delta and rho seem to be, if you will, all-terrain. When we start looking at other areas of the model, beta and C, beta and C, we get a somewhat nuanced answer. Generally speaking, therapies that promote variant kill, those are things like antibodies, right? Pulling the virus out of circulation and preventing it from going anywhere. Those seem to be better than simply inhibiting infection, so providing barriers to entry and preventing the virus from getting a foothold in the cell. Unless, of course, we're just focusing on the epithelial cells infected, right? And in that case, if that's our objective, then inhibiting infection seems to be just as good as promoting variant kill, right? But we do get a different answer depending on what we're trying to do. Finally, the column for kappa, which is delaying the transition from latently infected to actively productively infected, that seems to be a poor choice in any particular context. So that we wouldn't recommend anything that was trying to target that area of the viral life cycle. Next slide, please. Another way to think about these results is to consider that treatments are optimal only defined by a context. What are you trying to do, and when are you trying to do it? So on this plot, I've got the time on the x-axis, and the black line reflects the predicted variant count. So sort of going up, finally detectable around day 5 or day 6, peaking at day 9, and then falling off again through, say, detectable through, say, day 21. I've superimposed with that plot in the yellow and orange curve the healthy epithelial cell pool, which again starts intact at the beginning and then has this precipitous decline between day 6 and day 12 of the infection. So depending on where you are in time, you might target different portions of the viral life cycle. Early infection, perhaps day 0, 3, 6. The goal there might be to protect those healthy epithelial cells, right? And in that case, therapies targeting beta and C are superior to those combinations targeting delta and rho. But at some point, your goals need to change, because you can see from that golden yellow curve, we've really exhausted that healthy epithelial pool. There's no more of those left. So there's really not much benefit there to protecting that population. So there you might instead switch gears and go after targeting delta and rho and perhaps C. Next slide, please. Now, remaining few minutes, I'm going to walk you through some clear examples. Again, returning to the cell life cycle, looking at combinations that target delta and rho, which from our perspective seem to be kind of the workhorse or backbone choices. So that might be perhaps interferon and ribavirin to give you the sort of delta effect, and then perhaps a protease inhibitor or perhaps remdesivir to give you the rho effect in the model. If you then want to add something else on, you might add convalescent plasma or antibodies or ultimately a vaccine. That gives you the C effect on top of the other two components. So let's walk through those case examples relatively quickly. Next slide. I'm not going to spend any time here, but I will point you to the preprint where we go through a variety of different compounds, all the ones that we were able to identify, and kind of walk through which of these drugs seem to be targeting which portions of the viral life cycle. So if you're interested in that, I direct you to the preprint. Next slide. So let's look at some interventions at peak -3 days. So again, these subjects are relatively symptom-free at this moment. In the upper left panel, that's the virion simulation. Is again, to orient you, the black line is the null intervention, right? If we do nothing, this is what the viral trajectories look like. When we go in with single target agents that are relatively modest, so I mean by that, perhaps a 50% inhibition of something the virus is doing, or perhaps a doubling of something that we're doing to clear the virus. All of those single targets look to be, not that effective in terms of moving the viral load around. We do, in the upper right, we do spare uninfected cells, and clearly we're knocking 2 logs off of the reduction in that population. So that's certainly good. Moving to the bottom rows, now we start putting together 2, 3, and 4 targets together. Again, each one of these has a relatively modest effect, but then you can see this really dramatic change in the lower left plot, where all of a sudden we go from these curves that sort of go up and down again, and we actually break the viral infection pattern. It can get this really rapid resolution of the viral curve. And then when we look at the unaffected epithelial cells in the lower right, you can very clearly see that we're really sparing that population. Suzanne, let's jump ahead one more slide as well. That's great. I'm going to make back some time there. Great, thank you. So this is intervention at peak +3 days, right? So now we're talking about 12 days after infection. That upper left curve shows what happens with single targets. And with the exception of the single targets targeting delta, we're really not doing much in the way of affecting the viral load. And in the upper right panel, you can see that at this point, there isn't much to be done about the epithelial cell pool, right? Those are relatively exhausted. In the lower left, when we go in with multiple targets, we can clearly see that we start adding 2 and 3 of these drugs together, however modest. We start shaving a week or so off of the duration of viral shedding. and that we could be really important from a population transmission perspective. Next slide. So our conclusions. To date, no single agent has presented itself as capable of arresting the viral life cycle. Our hypothesis, we should take what we learned from the HIV pandemic and apply those concepts to move adroitly into combination therapy, hoping that we find targeting different segments of the life cycle to be more effective than the monotherapies alone. And this viral modeling tool is one such framework we can use to make quantitative assessments about what if A, what if B. Next slide. Some brief acknowledgments. I'd like to thank Jeremy Gage over at INSERM, Patrick Smith, Kasia Patel, and Jim Gallagher here at Certara, and of course Steve Kern and Dan Hartman for their continued support at the Bill and Melinda Gates Foundation. Next slide, and I'll turn it back over to Suzanne to wrap it up. Thank you very much for your attention and time.
Thank you so much, Mike and Ed and Rajesh. We got a ton of questions coming in from our audience, and that's fantastic. We'll answer as many as we can. So first question, someone wants to know, recently a study says that we can use a steroid for COVID patients on oxygen ventilators. How much of that is effective? So I guess they're talking about dexamethasone. And then there's a second part talking about hydroxychloroquine. We usually associate it with malaria. What do you think the status is as using it as a drug for COVID?
I can take the second part of that question, Suzanne. Is that okay?
Yep, that'd be great.
Okay, great. So I think it's a wonderful question. Thank you for the question. One of the parts of repurposing a molecule for a different indication or disease, it's actually not that uncommon. Interestingly, we talked about AZT in HIV. That AZT was actually first looked at for oncology before it, got effective in HIV. Anyway, that's a tangential point. But when way back in 2003, when the first coronavirus events took place with regard to both the SARS-CoV-2 as well as MERS, the Middle East Respiratory Syndrome, there was a number of studies done at that point looking at a lot of drugs. In fact, much of those drugs looked at that time are the ones that were, basically looked for the current COVID-19 situation. So I think one of the issues with taking a drug that was experimentally used and perhaps even seen to work in vitro is the uncertainty about how the compound works. So there's not a lot of detail on what the mechanism of action is for, either hydroxychloroquine or, for that matter, dexamethasone, the compound that you asked. So unless we understand through binding experiments, biochemical experiments, we will probably not understand the true mechanism of action of these compounds. And I think, and that's the part of the designer therapeutics that Mike was referring to, and maybe he can expand as well, is you need to have some biological plausibility when you expect a certain compound to work. And it's really not inconsistent with that philosophy that you actually see a lot of failed trials for compounds for which there was some underlying partial explanation for effect, but when you look at clinical studies, they seem to fall apart. I mean, I can try the first question, but maybe other panelists might be able to shed some light. But my understanding is the dexamethasone example, I don't recall seeing a peer-reviewed publication, so I don't have the full details to provide you more context to that particular question, but perhaps some of my panelists do.
Ed, do you want to comment on that?
Well, all I can really say about the DEXA findings are that it was reported from what has been quite an open trial, the recovery trial. It's UK-led from Oxford. It involves almost all of the NHS. It's a very large, simple platform trial, but they haven't released the results from any peer-reviewed environment. We've just had an announcement that it prevented 1 in 8 deaths amongst ventilated patients, I think it was. So it's very difficult for clinicians to make any recommendations based on that. And we don't know about what other treatments that the patients were receiving. So it was DEXA on top of whatever standard of care that they were receiving versus standard of care.
Thanks, Ed and Rajesh. Ed, I've got a question that looks to be directed towards you. Viral loads are usually measured in nasal swabs or saliva. How would you bridge the blood concentrations of a drug to effective concentrations in the respiratory epithelium?
I think it'd be better maybe if Rajesh answered that.
So I think I can provide you with some response, but I think we may have to take that one offline. So I think the fundamental is you're trying to assess effective concentrations. You would be able to discern that through, isolates where you incubate your compound with the, SARS-CoV-2, infected cell line, and you would find an IC50 value, which based on that, you're going to extrapolate to what might be a site of tissue, site of action tissue concentration. Of course, what you're going to do is use some kind of a physiologically based pharmacokinetic model to provide you with that precision around what that estimate is. Unfortunately, when you do not know experimentally how much of the drug is accumulated in a certain tissue, you'll have to make some assumptions and around the estimates, but that would be perhaps the the best way to understand site of action tissue concentrations. I don't know, Mike, if you wanted to share or add anything else.
Looks like we've got another question. Mike, feel free to chime in. Is the natural history consistent across the COVID-19 populations, i.e. mild versus moderate versus severe patients?
Ooh, tough question. The modeling work that I showed you previously is conditioned on, I think, around 14 subjects. In the next week or two, I think we're going to be updating that to maybe 48 subjects worth of data. So, you're really seeing this information real time. So I think it's very difficult to understand at this moment what degree of variability we see in the viral endpoint, other than it's pretty high even within a subject. When you take serial sampling within a single individual, you get a great deal of variation. So that then makes it very difficult for us to just understand, well, what is the variability in the population? So I think it's too early to comment on that for SARS-CoV-2. What we can say is when we look at other infectious diseases, and I'm thinking particularly about influenza, we do see, for example, in immunocompromised patients, very long durations of viral shedding. So we have a hint from other infectious agents, then yes, sure, you're highly likely to see different patterns of viral kinetics depending on what's going on in that particular subject. So great question, thank you.
One of our attendees wants to know, how are the viral load PCR copies normalized to volume, i.e. per mil, given that the sample is a nasal swab?
Ooh, tough question on the bioanalytical side of things. Yeah, so that information then has to be converted, if I recall correctly. That the actual viral swab information then has to be converted to to the copy amount, and we're assuming in the model that the volume of the epithelial lining is something like 30 or 40 milliliters. So we're sort of assuming a homogeneous space throughout the lung, where we're getting a sample that tells us something about that rough concentration over that entire volume.
I think this is another question for Mike or Rajesh. I would think that the defining markers for the clinical proxy would give a venue for modeling, but my guess is that the cytokine fingerprints would also be the candidates for this, since it potentially generates the mechanism of action link and the mechanism of interaction in terms of synergism and antagonism. Okay, I think that's just a comment. Okay. Someone else says there's variability in viral load from mild to severe COVID patients. Is the viral load still reasonable as a surrogate endpoint of treatment, 1? And 2, there's several factors that might be contributors for mortality in COVID-19. What's your opinion about targeting about the combinations that target these contributors?
Yeah. So on the first question, is the viral load still reasonable? I don't think that it is required that we somehow model viral load. Ultimately, the endpoint that's going to stick is mortality, right? That's the thing we care about. However, we found in more traditional drug development that if we can follow and describe and quantitate a causal chain of events, you treat with the drug, the virus level drops. When the virus level drops, symptoms abate. When symptoms abate, mortality declines. When you can start tying together stitching together that story, you start being able to get a richer understanding of what's going on and make better decisions around how we can modify treatment to get a little bit more effective mortality out of that. So yes, ultimately, we need to be looking at the final endpoints, but historically, we found these intermediate causal chain components worth describing.
Yeah, and oh, sorry, Mike.
Go ahead.
No, I wanted to add that from an early drug development perspective, that's what you have. If you have viral load data, that will help you pick the right dose. And one of the challenges with data as we are seeing rolling in is we are not able to discern effects, and that's something that we saw with remdesivir. We have seen with other agents. There is a practical and indistinguishable viral load profile between active drug and placebo. So I think from the perspective of early drug development, enriching your trial as much as you can with biomarkers can help you with dose selection, perhaps even trial design. But in terms of moving the needle on the disease, obviously the more distal endpoints, hospitalization, time to recovery, so on and so forth, might be more value added.
Looks like we have a few more questions. This is really great that our audience has so many questions for our speakers. There is viral shedding and COVID-19 infection. Could your model add and examine these factors?
Yeah, so we would think, although this model doesn't explicitly handle it, we would think that if you have detectable virus, so on those curves that I showed you, where you exceed something like 100 copies per mil, so you can detect the presence of virus in the qPCR technique. If you could detect that, possibly that those subjects then are infectious, right? They're also those droplets are shedding from their body and moving about the environment. Potentially the concentration, where you are on that y-axis might have something to do with the number of infectious particles that an individual is throwing off per minute. So we don't have we couldn't translate that y-axis to, I guess, degree of infectivity, but you might imagine that they're somehow proportional. I hope that helps.
This might be a question for Ed. what proportion of these trials are from China or Taiwan? There's research. What research is from these countries? What is it showing? They have the advantage of having dealt with previous coronavirus epidemics.
Thanks. So that's a great question. About a third of all trials are from China. And if you've been following the media about the Chinese epidemic, then you will know that it was the early stages of the epidemic or the pandemic were in China, and much less now, although there's an outbreak that appears to be in Beijing. So as many of you will know, when you set up a trial, it takes quite a bit of time to organize for funding, to go out, organize for IRBs, to get the drug, to get the swabs, etc. And so this was taking, several weeks to months. And so even though they were burning through the epidemic, they still were not recruiting. And so what we'll end up seeing from China, although they were ahead of everybody else, is still only a handful of trials will ever read out. And many of them are small, not great quality. And so I think about a third of trials are from China, which means about a third of the trials that have ever been registered, you shouldn't count on ever seeing. Because they won't recruit it. And the only solution to that is if we can convince people to hand over data to a global central database. And there's work on that, what's called the Workbench, the Alliance Workbench, which is hosted in the UK. Hopefully we'll get to obtaining data from around the world. But what I think we now need to think about as people in industry is, can you follow the epicenter of this pandemic? And so for example, right now I'm beginning recruitment of a trial in Brazil. Brazil is on fire with the epidemic right now. Who knows how long that's going to last? And then we're planning for trials in South Africa, because that's a place that we project is likely still on the way up. And I think industry needs to be thinking about not only where you can find groups you've worked with before and have access to different interventions, but where is that epidemic going to be when you really do have all of your approvals and drugs in place?
Thanks, Ed. Mike, someone wants to know about your opinion between the relationship between viral load, viral kinetics, and COVID-19 disease severity.
Well, that's another great question. So I would think perhaps in the first 3 weeks of the disease, it should not be taken as coincidental that when these subjects get to what we think of as a peak viral load, perhaps around day 9, that's when they're becoming symptomatic, right? Fevers, chills, cough, that sort of business. So, clearly the time, at least of the early symptoms that are showing up, align reasonably well with what we're seeing in the model. What we don't handle then is, well, according to the model, the epithelial cells are exhausted by about day 12, and the virus is kind of on its decline and on its way out by the time we get to day 21. So the model is completely silent about host response, right? ARDS, CRS, other organ involvement, stroke, etc. The model doesn't have anything to say about that. So we still got some distance to go to really talk about what happens in the host response portion of the disease. Perhaps this model is reasonable for capturing sort of the viral infection point or period of the disease, but it says nothing about what happens with the host response, which seems to be a major sequela for some of these, for some subjects who acquire this disease. So I guess in summary, I think the model handles things relatively well in the early timescale of infection, but it's silent about the long-term consequences, with the exception that it does track the exhaustion of the epithelial host cells, and I think that's an important component that people haven't spent a lot of time thinking about.
Can you comment on why the WHO is removing hydroxychloroquine from clinical trials rather than emphasizing the part of the cell cycle where the drug is effective?
Well, I'll answer the first part of that. I think the withdrawal of hydroxychloroquine from the solidarity arm was a legitimate decision that was made yesterday or the day before by the data monitoring committee. Previously, there had been a lot of media attention based on a fraudulent Lancet article which many of you are aware of, that indicated that hydroxychloroquine may be causing greater death than in those who don't receive it in the hospitalized population. That was the Lancet now withdrawn article, a retracted article. The WHO at that time made an announcement they were putting their hydroxychloroquine arm of the Solidarity hospitalized patients on hold. Then they put it back on. And then from my understanding, there was a data monitoring committee meeting in the last few days that looked at the data and concluded that it was unlikely to show any treatment benefit. Perhaps it showed the harm. They haven't released the data except to say definitely doesn't exhibit a treatment benefit in that population. And for that reason, they were stopping their arm.
Ed, this might be a question for you. Can you comment on the impact of gender and age on the mortality rate?
Well, I mean, I'm not sure how much I can comment on it for the purpose of completed clinical trials, because there have been so few clinical trials where you can get access to the actual data or read the final publication that describes anything. What we do know is that males have a greater rate of death from epidemiological evaluations, and that older age is strongly associated with mortality. Of course, older age is strongly associated with mortality at the best of times, and male death is always associated with greater rates of mortality, even at the best of times. So we don't know if this is driven by the virus or if this is simply, exacerbating underlying conditions and risk factors, and risky behaviors that result in greater risk factors.
This looks to be the last question. A very large number of trials in China show traditional Chinese medicine as the intervention. It's a good number on the CITEL tracker. Can you speculate where this type of intervention might interfere on the viral kinetic model you showed?
Tough question. I have to admit this showcases a big blind spot in Western medicine, because we don't have a good mechanism of understanding around what a lot of these compounds do. I think you would probably go about answering that question, which let me preface that with I can't do that. But I think you would go about answering that question the same way we did in the preprint. You sit down with a list of compounds, you look at their mechanism of action, and then you try to understand, well, where would that mechanism map to this viral life cycle model? So again, I think that same process would apply here, but you'd need someone that was well versed in the mechanism of how these traditional Chinese medicines work.
All right. Thank you so much, Ed and Rajesh and Mike, for that excellent presentation, and thank you to the audience for all the wonderful questions you asked. Just got a few short announcements before we end the webinar. Our next webinar will be next Thursday, June 25th at 10 a.m. Eastern, where we will be talking about accelerating vaccine development, application to COVID-19, and future pandemics. You can visit certara.com to register. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.